Assalamu alaikum wa rahmatullah. In the previous video, I have discussed this topic, which is about the assessing the accuracy of the docking, and this by comparing the crystal pose with the docking pose, and then measuring the root mean square deviation. If the value less than 2 angstrom, so that means your docking is good. So that means the smaller the value of the RMSD or root mean square deviation, the accurate the docking. As I told you that I will show you a tutorial or run for this, or how this can be done using MOE. Okay, I will do this, but before this, I have to complete the following few slides. Okay, so the next one is about preparing the ligands. So before molecular docking, I have to check my ligands. Okay, so you know that the ligands are obtained by sketching the ligand using any of the sketching software like ChemDraw or ChemSketch. Also, we can get the ligand structure from web databases such as PubChem database or Zinc database. From these databases, we can download the 3D structure of the ligand as SDF file, or we can obtain the SMILES code or SMILES notation of the ligand, then paste it into the software, which will convert it into 3D structure of that ligand. So I think this one of the best methods for obtaining the 3D structure of the compounds by taking the canonical SMILES from the database, then just you place it into the software, and the software is going to convert this SMILES into 3D structure. Okay, so these are the different methods for obtaining the 3D structure of the ligand. A reasonable 3D structure is required as a starting point. So I told you how to do this, or how to obtain this 3D structure. Even during flexible docking, bond lengths and angles are held fixed. You know, bond angles and bond lengths held fixed. They are not going to be changed. Okay, but you know, only the flexible bonds or sigma bonds will be changed during energy minimization. So there will be a rotation about sigma bond. The protonation state and tautomeric form of a particular ligand could influence its hydrogen bonding ability. So you know, tautomerism, mainly or especially the keto-enol tautomerism, this is very important because there is interchangeable two interchangeable structures. So that means the structure could be shown in two different ways. So this is an enol, en for the double bond, ol for the OH. So this tautomerism or keto-enol tautomerism. So this compound or compounds containing such a functional group exist in one of these or both these structures. So look at this here. The protonation state and tautomeric form of a particular ligand could influence hydrogen bonding ability. So look here, this is hydrogen bond acceptor, okay, because of the, and here we have a, yes, this is hydrogen bond donor, and also this can accept hydrogen bond, so this hydrogen bond acceptor. Okay, so two hydrogen bonds could be obtained from, I mean two sources of hydrogen bonds are present here. either protonate as expected from a physiological pH. So this depending on the pH of the site of action. Depending on this, you can protonate or leave it as it is. Okay, so we can, for example, if your compound has the possibility to exist as enol or ketone, so in this case you can dock both structures and then you can compare the score. So, or generate and dock all possible protonation state and tautomers and retain the one with high score. So what we mean by this, if you know the pH of the site of action of the compound, you can just dock one of these depending on the pH value, if it is acidic or basic. If you have no idea, you can dock both. Dock this one and dock this one, and then you compare the score. You just take the compound that give you the highest score. Preparing the protein structure. The protein data bank is a repository of a crystal structure, often in complexes with inhibitors. So as I told you, when you download protein from protein data bank, this protein has a ligand attached to it or bound to its it is complexed with this protein and found in its binding site. PDB structure often contain water molecule, and this is normal because when crystallizing the protein, this is done in aqueous medium. Okay, in mainly it is aqueous. Okay, using water. So sometimes water is crystallized. Okay, water of crystallization is obtained or found with the crystal structure of the protein. In general, all water molecules are removed. So in normal molecular docking, we remove water molecules. But if from the literature you know that water molecules in the binding site play an important role in coordinating to the ligand, so you have to leave those water molecules. So for example, assume that this is the binding site, okay, and this your compound here. Yeah, just assumption. So this is like we have here OH here like this, and here we have amino acid which has, for example, let's say C double bond O. If you know that water can form like this hydrogen bond here and then hydrogen bond here. So water contributes the interaction. So water act as bridge between the amino acid, okay, and my ligand. So such water molecule is important for binding. So if you know something like this, keep it. Do not delete this water molecule. Do not remove it. Okay, but you can remove solvation water from the rest of the molecule or the rest of the enzyme, but leave these water molecules because they play important role in coordinating with the ligand. Okay, this is what I mean by this. Okay. And this can be done easily using the docking software. It's not difficult. PDB structures are missing all hydrogens. Yeah, this because X-ray crystallography cannot recognize hydrogen atoms. So protein structure from X-ray crystallography lacks hydrogen atoms. So in this case, you have to add all these hydrogens. Many docking programs require the protein to have explicit hydrogens. So hydrogens will be added will be added because or you have or must be added because hydrogens are very important for hydrogen bonding. Okay, for hydrogen bonds. Okay, so hydrogens are very important, mainly the polar hydrogens. Okay, polar hydrogens are very important for hydrogen bonding. So that means before I perform any docking, I have to add hydrogens to my protein. In general, these can be added ambiguously. Okay, ambiguously, except in the case of acidic basic side chains. Okay, because sometimes yeah, in all of the proteins or enzymes, there are acidic and basic side chains. So in this case, you have to check, and some software can allow the accurate addition of hydrogens and addition or removal of hydrogens from these residues or amino acids. An incorrect assignment of protonation state in the active site will give poor result. For example, glutamate, aspartate have carboxyl. Okay, so this deprotonated and this is protonated, and you know that OH is hydrogen bond donor, O is not. So here this is not going to accept hydrogen bonding, but in this case if it is protonated, this will give higher possibility for forming hydrogen bonds. Okay, also basic amino acids such as histidine, okay, have two tautomers like this. So you look at this nitrogen. This is unprotonated, deprotonated, and this protonated. So this hydrogen can travel to here. Okay, so this can generate this cation. Okay, so here this has higher possibility for forming hydrogen bonds because of the presence of the hydrogen, two hydrogens, this hydrogen and this hydrogen. So this is a protonated form, and it has a good chance to form two hydrogen bonds this way and this way. Okay, so yeah, this is the end of our talk concerning molecular docking. Now let me show you how can I perform molecular docking for the co-crystallized ligand, and to see the root mean square deviation value in order to check or assess the accuracy of molecular docking process. Okay, so I will show you this now. Okay, this is MOE software. Okay, I will use PDB structure of one of the enzymes saved here in the tutorials of the program. Okay, sample, then mol, then this is my enzyme. Okay, yes, you can see the 3D structure of this enzyme. Okay, I can use this, can rotate this structure. Okay, this protein has no hydrogens added to it. I click sequence to see the sequence here. Here you can see, if you look here, these are the amino acids: serine, isoleucine, serine, arginine, phenylalanine, and so on. This zinc molecule, and this is the inhibitor. This ROL, this is the inhibitor or the co-crystallized ligand. These are three water molecules. I can keep them or delete them because here my concern is just to run docking and to check the accuracy of the process. Okay, but let me delete these hydrogens. Okay, or just leave it. Okay, leave it. Okay, now let us add hydrogens. Compute protonate 3D. Okay, so this will add hydrogens. You can see hydrogens has been added. Okay, uh, this will take like a few seconds. Okay, the protein structure is not that big. It is small. Okay, so in this process, hydrogens are added and the energy is optimized. Okay. Okay, now protonation is done. Let me show you the binding site of the protein, surface and maps. Okay, apply. Yes, when I apply this, there is here, you can see this cavity. Okay, and then isolate. This will hide everything except the binding site. And also I can make it a little bit clear by removing backbone. Okay, now let me show it a little bit bigger. Okay, rotate. Okay, now can you see that here? Um, this is the ligand inside the binary. Now let me zoom in this. Okay, now I can see. Yeah, this is my ligand. This ligand is the co-crystallized ligand. This is the co-crystallized ligand. Okay. Now I will perform molecular docking for this ligand. Okay. I will redock it again and compare the docking. This is what we call this now. This is the crystal pose. Crystal pose. Now I will perform molecular docking to see the docking pose. Simulation dock. Okay, here the output, you save your docking anywhere, doc 1, let's name it doc 1. This is the output file which in which the docking result will be saved. This is a database, okay? And a receptor, I will keep it as it is. Site, ligand site. As I told you, the software use this ligand or co-crystallized ligand in order to or use it for directing the ligands from any database. So because of this, we set site into ligand atoms. So that means the binding site will be defined by the ligand atoms. And also here, ligand, what what is going to be docked is the ligand atom. So that means these atoms will be redocked. If I want to dock a database, I open this and I go, I click MDB molecular database, and then I go to browse and until I get my database. But now here I just want to dock this ligand. Okay, here these are the docking parameters. Okay, the search algorithm and the scoring function and so on. Yeah, I use default some of the default values. So here placement, I use triangle matcher. This as told by the tutorial. So according to the tutorial. So pre-scoring, I use London DG. So this is the scoring function. Here return, as I told you, that the scoring function generates different poses. So here the poses are 30, but for saving time, I will just change it into 10. So that means the software will generate 10 poses for this. Look at this, remove the duplicates. If I got duplicate results, so this will be removed. Okay, sometimes I obtain less than 10 results. Refinement, so this will be done by force field, and rescoring will be done by London DG, and I will return this to 10, and then okay, and then I click OK. Okay. Okay, so this will perform the docking. Now you can see the compound is being docked. Now you can see the compound is moving. Okay, it is vibrating here. So these are the different poses. You can see the movement of the compound. The crystal pose is stable, but these are the docking poses. They are moving, and this table after the docking is finished, it will show the binding energy and root mean square deviation values. Okay. You can see here the ligand tries to find orientation similar to the crystal pose. Okay. This should be faster than this. So this depending on the computer you are using, the time will be taken for if you are using a faster computer. This will take like few seconds. Okay, but I think I have processing problem here. Okay, now you can see these are my results. Okay, this is the same ligand. So look at, I got 9 results instead of 10 because one has been omitted or excluded. Okay, S, this is the scores. These are the binding energy in kilocalorie per mole. And here these are the RMSD, root mean square deviation. Look at the first 3 readings. I got here 0.93, here 0.81, here 0.4. So I think this is the best one because the value is small. I told you that the cutoff value is 2. So, and up to this has good, but the following have higher root mean square deviation, RMSD values. So now let's compare this structure by placing the docking poses on this crystal pose. I will show you this now. Okay, let me move this. No, yes, okay. Now, I will remove this surface because I want to see the compound clearly. Okay, clear. Okay, so now I will color this using a different color. For example, I'll give it green color. Okay, now I will open my docking results. Okay, browser. Okay, then I will change the color of these compounds into yellow. Okay, I would change into sticks. Okay, then I will remove the hydrogens in order to see the compound clearly. Okay, now so this is the superposition of compound 1, this compound 1 which has energy minus 12.1. It looks nice, a little bit. Yeah, this ring, those 2 rings are overlapped in a very nice way. Okay, now let's check compound number 2, which has smaller root mean. Oh, very nice. So look at these are totally superimposed, and those are superimposed, but here they are a little bit different or a little bit unsuperimpose. Okay, and now let's check this, the smallest RMSD. Now this is the perfect one. This is the perfect. I think here we obtain a very nice overlapping. Okay, for the 2 compounds. The compound in green, this is the docking pose, and the compound, sorry, this is the crystal pose, and the compound in yellow, this is the docking point. So that means the docking could generate or could give a result that's similar to the experimental one, because the compound in green is an experimental or obtained from experiment, and the compound in yellow that is obtained from docking. So that means a molecular docking process can predict the pose that which is similar to that of the crystal pose from X-ray crystallography. Okay, I think this the end of my lecture. I hope that you get the maximum or the benefit from this lecture. Okay, thank you so much, and see you later.